You can see that, right? So yeah, the first question was select the incorrect state just to add. Bright microscopy can only provide morphological details and not quantify chemical height and mechanical and physical properties. That's true, right? Bright microscopy does not do that. It's a simple, most simple form of microscopy. It just allows you to look at the overall morphology. So that's a correct statement. And then second was samples of the left-hand class are better observed through bright field than dark field. That is the incorrect answer. So which means that's the right choice for that question, which many of you did get, which is good. And the other three, DIC, which is also correct because the images are not topologically accurate. It's a big perception of difference in optical path or optical path difference between various sample constituents. That's also the answer. I mean, it's correct. It's a correct statement. And polarization microscopy renders transparent object into fascinating, colorful object because it's mostly used to observe crystals and like geological, like stones and things like that. Bacteria are not usually, and depending on which direction the light enters, it makes it look colorful. That's like a normal observation with polarization microscopy. And fluorescence microscopy allows imaging structural components and determination when you tag it with a specific fluorescent tag. I remember I did mention that DAC allows you to see the 3D topography. Do you remember that? Yeah, you get a little bit more. You can see the depth. The bigger part of the sample, you get more interference or whatever. So you'd be able to see sort of difference between the two. And so is that what you mean by topographically accurate? It enhance a little bit to amplify the difference of picture? Exactly. It's just making it look like 3D, but it's not accurate. And when you compare this to AFM, for example, AFM or even SEM, it also allows you to see the topography, but in AFM, also in a sense, let's not complicate that. So when you look at AFM, it's accurate Z distance that you're seeing. Z means light. Not the distortion of the visual. That's exactly right. So it's not accurate, it's inaccurate. It's a fake perception of 3D. It's not quantifiable. It's just making it more real, but that's fake. Yeah, sorry, the DIC is a good representation. That's right, yeah. Oh, I can't hear them? Can't I hear? We can't hear you. Okay, well, I should put my laptop on mute.
Are you here?
Now I can, yes, but that might be through my laptop. I am. Now I can hear you. Yes, sorry about that. I don't think things always go sideways with Zoom. But yes, now I can hear you. Did you have a question? Sorry.
We were asking a few questions earlier, but you couldn't hear us.
Yeah, sorry about that. I can hear you now, but it might be through my laptop, but that's okay. I can still hear you. Go ahead.
We were just saying we can't hear what people are asking you in class. People were asking you, you said something about your office and something helping something. We can't hear anyone else.
Yes, no, fair enough. So somebody here in the class was asking if if you are able to get your answer booklets back. And then I said that you were welcome to drop by my office and then go through it if you want, and then you'll have to leave the answer booklet back with me. And there were a couple of questions about the first question in the exam about the DAC. Thomas here was asking like that the C option for the first multiple choice was the most confusing one, because he was just trying to get a clarity on what the fake 3D appearance on the DAC microscopy was. I think that was it. I don't think we discussed anything else.
And then I think that Jade said that it would be helpful to go through the answers after we have our midterms back. So if you could post it. I don't know if you heard her say that.
It would be helpful to go through what, sorry?
Well, you're going through the exam right now, but
Yeah.
we haven't gotten our marks back to see what, to see what we got. So it's just, it'd be nicer if we had access to what we got on it before we went through it, so we knew where we stood as to how much to write down right now.
I feel like it'll be helpful either way, don't you think? If you got your marks, I mean, yeah, you're welcome to drop by my office and see where you went wrong. Like I would be only giving you your total points, not how much you score in each of them. And you know
Okay.
how you answered each of those questions, right? Does that make sense?
Yeah, I think she just wants you to post the answers so that she can go through it once she gets her mark.
Post the answers. I may not be doing that.
Yeah, no, it makes sense now. I didn't realize you weren't giving us what we got, like which questions we got right. You're just giving us our total.
Yeah, I'm just giving you your total. Yeah, not for each. And then again, like I said, you're welcome to drop by my office, and then I'm happy to, like, once you go through your answers, I'm happy to, like, can explain if you want, like, where you went wrong and what would have been a better. And again, me going through your answers will also, you'll kind of have an idea when you look at your answers. Okay, so the second, is the echo bad? Or is, okay, it's not bad. Which of the following methods can be used to measure bacterial cell counts in a chicken salad? I thought this may have been a very straightforward question, but many of you have stumbled on this one. And the correct answer is serial dilution and pour plate. And I've given you points even if you put MPN, because they both contain serial dilution. So if you answer either of them and give me some sort of a justification, I give you full points for that. And many of you have chosen hemocytometer, which I was kind of disappointed with, because chicken salad, it's a pretty complex, I mean, if you think about the food, there is lots of things going on in chicken salad, right? Like, how could you possibly visually distinguish bacterial cells from the food? It would be impossible.
I didn't think, like, are you trying to get at the Reese's chicken salad in a liquid and dilute that in a broth and then plate that?
Okay, I see. Yeah. Yeah, when I say serial dilution, you will take a portion of the chicken salad. You don't have to grow it. You can directly serial dilute it, because that's how you measure bacterial load in a soil sample, for example, right? And it's a very commonly used technique in microbiology. Feces, any type of sample you have.
Small section of, say, that chicken salad, and then are you extrapolating it to the whole salad then once you've done your serial dilution?
You homogenize it, ideally. Like, you blend it up, and then you take a small sample of that, and then you serially dilute it, and then you plate it. And then you extrapolate back, like you weigh like 1 gram of chicken salad, for example. And then you serially dilute it, you plate it, and then count how many colonies you got. And then you would display your number using CFU per gram of the chicken salad. That's the total number, yeah. Yeah. And number 3, select the correct statement. most of you got the correct answer for this one, and answer is E, because all the other ones are incorrect, because volatile granules are metachromatic because they absorb bright stains heavily. That's not true. That's not why they're called metachromatic, because it changes the color of the stain cell. That's why they're called metachromatic. And the second option, prokaryotic chromosome, is haploid, linear, except for plasmid. It's kind of made it tricky that way because only one item of that is wrong, and that's where it says chromosomes are linear. Bacterial chromosomes are not linear, they're circular. And option C, many of you have picked this one, which I kind of understand, because there are exceptions to some bacteria that don't have peptoglycan, and I was hoping you'd pick up on that. And then D, it's a fairly straightforward one because energy of the ejected electrons is always less, or in other words, the wavelength is the one that's higher compared to the excitation wavelength. And so the answer is E, which is correct. And then fourth one, you're given a sample of scrapings from a rock with a thick biofilm from deep sea, and then there are diatoms in it. And how would you?
Down, we cannot see question number 4.
So the diatoms embedded in the biofilm, and what type of microscopy you would use to specifically morphologically characterize the diatoms. So you have to sometimes pay careful attention to what we are trying to address. So like I said, the ideal technique that I would use based on my microscopy experience would be confocal. Because confocal has a pinhole that ejects out of focus light, and it has the ability to focus on different levels along the Z dimension. And so it's called image slicing, which allows you to focus on different levels because the diatoms are embedded at different levels. You would be able to focus on the diatoms themselves and able to morphologically characterize them. But if you didn't know those type of details, maybe you picked polarization microscopy, because polarization microscopy is the ideal technique to image diatoms because of their cell wall, the crystalline nature of the cell wall. And that would be a correct answer too, but then you have to justify. Some of you have picked like AFM. AFM would be the worst technique to image diatoms embedded inside, because AFM is a surface imaging technique, right? The diatoms are embedded inside the biofilm, in which case it would be almost impossible to see what's inside. And same thing with the SEM. SEM will let you image anything that's on the surface, but not at the depth within the sample, right? Does that make sense? And then dark field.
If we choose any of those, they're correct.
Sorry?
If we choose the ones you mentioned above, we will be correct, because
No, you would not be correct. I was really generous in giving you grades this time. If you showed me some sort of a justification of how you would do it, then I have given you 1 or 2 points, but those are not correct answers.
But
like if you chose, like
in my notes, when you were teaching this, I have written it down. You said specifically for biofilms to see their characteristics and their morphological things, we would use TEM. And so that's why I think most students chooses the correct answers, because honestly, when you teach the lectures, it turns out to be like, you say things which are like, in generally, it's like accumulated, but then when you ask questions in the midterm, they were very specific. Because right now, you gave us multiple choice from A to G, and most of them are correct if you were to, like, honestly speaking, if you were to choose, because not most of them, but like SEM, TEM, and confocal, like these were the main ones, because I have clearly written it down on my notes when you were teaching that. So that's why I'm just confused at. So will we get partial marks, or it will be like?
Yeah, like I said, I did give you partial marks,
okay.
But TEM, I did give you partial marks because it does take a slice through your biofilm, and then you would be seeing some atoms. But SEM, is not a good technique. Like I said, it only measures the surface. I would have said SEM is ideal technique to image the 3D characteristic of biofilms, but not if something is embedded inside the biofilm. Does that make sense? So you could use TEM, but it's not the ideal. So if you did a good job of, say, you picked TEM and you did a good justification, I did give you points for that.
I think also a lot of people don't, I don't think it was super explained to us the difference between like morphological characteristics and topology and stuff and topographical stuff like that. Like, I don't think we went through that. Like, I took a microscopy class, so I went through that. But this question's like morphological characteristics of diatoms within a biofilm. I'm wondering, like, what are you looking for? Do I need the shape of it? Like, do I need the characteristics of the surface? Do I need the whole biofilm together? So I talked to a lot of people after the class and I was like, I'm not really understanding what she's asking for morphological characteristics, because I don't know if we really went through morphological, topological, shape, like what those questions.
I did use those terms in the class, and if something is not clear, like I don't know where you stand sometimes. It's hard for me to judge what terms are easy for you to understand, what terms are not. So morphology and topography means they're different. So morphology means overall shape. Topography means just the surface topography. And those are also used in common layman terms, right? Topological. I mean, fair enough that you didn't know exactly what I was asking. You could always ask for clarification because I don't know what you understand and what you don't understand, okay? Normal light microscopy would, well, it depends. It depends on what part of the biofilm you're focusing. If the biofilm is fairly thick, you'll get a bunch of fuzzy images. You won't be able to see like what's underneath. Like you might try to focus it at different levels, but then there's not going to be a lot of contrast. There's might be a lot happening in a biofilm. You won't be able to distinguish, say, diatom from some other organism floating around. Could be tricky. It would be, yeah, it would be part of the fluorescence microscopy because confocal is a type of fluorescence microscopy. And then I would have talked as in, under fluorescence microscopy. Yeah, it wasn't like a separate slide with a separate. No, I didn't make a slide for separate for confocal. It would be under fluorescence. But I did talk about it. Yeah. Okay. So question 5, which of the following structure is not likely derived from an endosymbiotic bacterium? The correct answer would be if you answered Golgi complex or plasma membrane, which most of you did get the correct answer.
Sorry,
so
before
that
you,
was fairly straightforward.
sorry to cut you off. I'm just confused what the correct answer for 4 ended up being.
For question 5?
4.
4, confocal is the best answer.
Is the best, but which ones did you accept for then?
I accepted polarization, I accepted TEM. I think bright field, I did give you part points.
Okay.
And then if you selected scanning electron or AFM without any justification, that would be a wrong answer. And I think I also give you part points for dark field.
Okay. Thank you. Sorry.
Yeah, that's okay. So yeah, question 5 was fairly straightforward. Question 6, yeah, that was just free grades for you. But then some of you just selected an answer without any justification. You could have written one sentence. I was just trying to get your perspective on how the class is going in general, but that's okay. So section 2, the first one. What do we
I don't think
treat
you're
IN
scrolling anymore.
Sorry?
We're still seeing just question 5.
Oh, yes, sorry. I think most of you, I don't think one person in the whole class selected all of them right, which is understandable. It was trickier than I expected. So first one should be straightforward. Fluorescence, I think most of you got it. Is polarization, I think most of you got that one as well. Bright field, C is fairly straightforward, but D, I thought it's easier than, it's AFM. And I thought it was easy to identify that. Many people had issues with that. E is phase contrast, which should be easy because there is a halo around the cells, which should be an easy indication that it's phase contrast. Many of you did get it. F is DIC, again, that fake perception of 3D. G, which I'm surprised with, many of you didn't get it, it's a dark field. So you can see the background is dark. The cell is looking bright. That should have been an easy thing to guess. And H was also phase contrast. Again, they have halos. 2 phase contrast, and the rest of them were.
Can you just
different.
repeat all those answers again? You just went through them a bit quick.
Yeah, A is fluorescence. B is polarization. C, bright field. D was AFM. E is phase contrast. F is DIC. G is dark field, and H is phase contrast again.
Thank you.
Yeah. What causes the halo is a good question. Mm-hmm. 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 Yeah. Yeah, no, that's okay. I think if I remember it correctly, you will get the most constructive interference near the edges, the boundary of the cell, because that's where the light would get diffracted the most, right? So that's when you get the set of rays that get most diffracted versus the ones that do get least diffracted, and that is constructive interference. That's why the intensity is much higher around the boundaries, and that's what makes it brighter. Yeah, yeah, yeah. Yeah, I remember mentioning it, that there's always a halo in phase contrast. Explain the reasons for placing archaea and eukaryotes in the same domain. I did talk about it. I thought it was a pretty straightforward, and I think most of you were able to answer that. Name 4 methods discussed in the class that can be used to differentiate bacterial species. Like, I didn't have a separate section in my lectures where I said, oh, this is how you differentiate bacterial species. I was expecting to kind of gather what you learned from all the lectures and just come up with answers. Like, you can use microscopy, you can use, in fact, I have some slides, like some example answers where some of you, I can probably show you here. No, do I have? No, I don't have for that one, but basically microscopy, some of you have just listed like all the different type of staining. I've given you points, but I did not say staining specifically. You could have used staining, you could use fish, like the fluorescence in situ hybridization technique. You could use microscopy, you could use other biochemical characteristics, catalase test. If you'd mentioned any 4 of them, I've given you points, and that's all I was expecting.
I have a question.
Sure.
Can we go back to the polarization ones?
Polarization, the question 1.
Sorry, just the pictures, not polarization, the microscopy
But
pictures.
yeah, sure.
Doesn't polarization microscopy often also have a black background? I think that picture you showed us of diatoms, or usually when you look at diatoms with polarization, it also has a black background.
For polarization, the background is not important, and it's not necessarily dark. That's not like a characteristic. For dark fields, for example, the background is always dark. For polarization, that's not necessarily the case.
Okay.
Yeah,
I think it's just that all the polarization microscopy I've seen, especially with diatoms, just looks a lot like that one.
It makes sense to have it dark because your sample looks brighter and colorful. When you adjust the contrast on your microscope, it will make the background darker, but it doesn't have to be that way.
Okay, and then do we have a picture of confocal microscopy on our slides somewhere? Does anyone know? Because I can't find it.
I don't think there's any slide saying specific about confocal. I think she just mentioned it in the class.
No, I talked about confocal under fluorescence microscopy because it's one of the types of fluorescence microscopy. So for confocal, the instrumentation of confocal would be same as a regular fluorescence microscopy. Fluorescence microscopy is a huge field in itself, and it's got so many different types of microscopies. Confocal is like kind of the main and the commonly used one. So I would have spoken at fluorescence, keeping confocal in mind, and I do remember talking about confocal in the class. So the instrumentation would also be applicable to confocal. Does that make sense?
Yeah, sorry, I'm just really confused. Do you mind putting the pictures of the microscopy up one more time?
You're asking me?
Yeah, sorry.
What do you mean by picture of the microscopy?
Like the question with the microscopy picture.
Oh,
Sorry.
yeah.
Which one was F?
I'm sorry?
Which was F?
Which one was F? F is DIC.
Okay, and then which one is C?
C is bright field.
Okay.
The class is going to be just talking about this. Okay, so I'm just going to quickly go through it. And if you have more specific things, like just drop by my office. So I'm just in the kinesiology second floor where the medical clinic used to be. So I'm 227 point, but just down the hallway. My office has my name on it. So just walk in from the front door to your right and just in the hallway where the medical clinic used to be, right across from the hallway, the same. So there's gym on one side and the other side is the research office. So just, yeah, send me an email if you want to come see the answers, but let's move on.
You have Zoom office hours at 2:30 today too, don't you?
I do, yes.
Yeah, okay, thank you.
Indicate 4 reasons why an antibiotic may prove ineffective against a pathogen. So again, this was like I talked about so many times. Again, you have to gather your knowledge and just answer this question. I will, pretty sure I have some answers. Yeah, so this here on the left side is actually a good answer. And many of you were able to answer this because it's still a fairly straightforward answer. So the bacteria might be forming a biofilm, the antibiotic may not, the membrane may not be permeable to the antibiotic, or the bacteria simply may have acquired resistance, but when you through acquired antibiotic resistance, which is basically taking up a plasmid containing a resistant gene, or it may be producing a pump, basically a membrane pump that's exporting the antibiotic to the external environment. And many of you did get answer for this, and I think it was fairly straightforward to answer. Why emission wavelength is always higher than excitation wavelength. Again, that was, I talked about Stokes shift in the class. We went through Jablonski diagram. You should have been able to answer. So the one on the left here is like the most precise answer. Basically, Stokes shift is the answer. But if you didn't mention Stokes shift, that's okay. If you'd said some of the energy gets lost through vibration or through heat, which is why the emitted wavelength is always higher or less energy. If you'd mentioned anything remotely in that zone, I've given you points. And somebody has drawn a figure here, which is obviously the best. You don't have to sometimes write anything. You can just make a figure. Pretty straightforward. Should have been easy to answer that one. Define the unique features of mitochondria compared to other eukaryotic cell organelles. I talked extensively about mitochondria, like how they fuse, genetic complementation, oxidative phosphorylation, how they have their own 70th chromosome, endosymbiotic theory. If you mention any of these, because it has 3 points, if you mention 3 points, 3 of those features that I mentioned, you would have got full points for that. Number 7, you are in the lab making a gram stain of E. coli. Like, I thought this would be fairly easy. I mean, you did have to show some thinking, but I thought it would be pretty easy to answer. What happened, assuming the error is in your technique? It was in bold. The problem is in your technique, not in the culture. So if you didn't focus your microscope, or if you put ethanol for too long, or because you're not seeing any cells, so likely you didn't focus your microscope, or you put in ethanol too much, so the decolorizing agent basically, so there was no stain, you forgot to put safranin. Anything of that sort would have given you points. You redo your gram stain and observe randomly arranged purple rods. Many of you said, oh, there's contamination. It's not E. coli. I don't think many of you even knew that E. coli is gram negative, which is, I don't know, I thought that was common knowledge at this point that you should know E. coli is gram negative. Maybe you didn't know that, which again is hard because I don't know that you don't know that. And you said it's contamination. It's clearly said the error is in your technique and not in the culture. You should keep that in mind when you answer your question. And the eighth question, a brewery in Regina has been facing a challenge. I mean, this type of challenges is what you will face when you move on to, in your career. I thought this is like a fun question. I don't know, I had a fun time coming up with that question. You basically had to take sample and then just culture it, serial dilution, see what's in it. Like any thought of thinking. I think there was a good answer here. Oh, I mean, you don't have to write much, right? Just take a sample, examine the process. Like, are they putting enough nutrients? Are they, like, what's different going on? What's not fermenting? The, yeast is what used in making beer. Even if you didn't know that, I was not expecting you to mention that it's the yeast. There was something problem with the yeast. If you had said, oh, there's contamination. How do you identify contamination? Microscopy, staining, biochemical techniques, any of the things. And number 9, like many of you complained that I asked this question because you didn't know that what's bacteriostatic and bactericidal, which may have been a fault on my side because I just, again, assumed that you knew that. It's hard to, I mean, it's easier to guess, I feel like, bacteriostatic versus bactericidal, but maybe if you didn't know. But like many of you were able to answer this question too. All I was asking is 2 graphs because this question was less about the antibiotic. It was more about the growth curve that we went through in the class. So when something is bacteriostatic, it means it's not growing. It's just static. It's not dying either. I mean, eventually they will die, which is why the curve will go down eventually, but that it has a long extended stationary phase where they just don't grow. And then the second one is bactericidal, which means the antibiotic is killing the bacteria. At the moment you add antibiotic, basically the cell number go down. And then many of you did make the graphs, but then you didn't label the X and Y axis. And then some of you have the graph going up. I mean, I don't understand if you don't label the axes, like what you're referring to. I mean, the graph going up could make sense if you label your Y-axis saying that, oh, that's cell death versus time or whatever. Is that good? So yeah,
I have a
will fix
question.
that your courses to make sure that you get your grades as soon as possible.
I have a question about this one.
Sure.
I think a lot of people I talked to that knew what they were doing on this is because they learned it in another class.
I'm sorry?
A lot of people learned this in another class. So I've talked to quite a few people who knew what they were doing on this question only because they had learned it before.
Yeah.
And
Yeah,
we never went over this ever. I've never heard these terms in my life.
No, fair enough.
And a lot of people I talk to have never heard the terms in their life. So
yeah,
people who are getting this right, they learned it before.
No, I get that. I don't know how to address that problem, to be honest, because I'm in a tricky situation too. I mean, I expect you're in your third year, fourth year, like you should know these terms, but you don't. When I'm drafting these questions, I'm always like, now I'm even more concerned because I didn't know that many of you had this issue.
Maybe these are more focused on like in biology classes than other classes. Like I'm a biochemist and I've never learned this in any of my biochem classes.
Biochemistry. Well,
yeah, maybe it's just like different focuses in like the classes we take. So like I'm in biochem, so we don't really focus on like the bacteria. So like I've never heard those words until they popped up on an exam.
Okay, yeah, I mean, I understand. Fair enough.
We can send you the prereq notes if you want. We can get some people together and send you our notes for the prereq for the class. And then that would probably be, because we're, it's fair to test us on anything we took for the prereq.
Mm-hmm. Yeah, so what I will do is next time, maybe when I draft my exam, I'll think about all the terms that we did go through in the class and not include the ones that I didn't even mention. Maybe that's just the way to go, because if I, get on this course of trying to go through what each of you took courses, I mean, I don't think that's going to work for me because I cannot remember, or like it's going to be a lot of work for me.
I just think maybe if you just don't assume that we know things and only test us on the stuff you teach us would be more fair.
Yeah, that's what I meant. Yeah, fair enough.
So are you keeping this question like to be marked on?
Sorry, say that again.
Are you keeping this question to be marked on, or are you just taking it out? Seeing as
I changed the total to 55. I am not taking out anything specifically.
Okay.
Yeah.
Okay. Sorry, we took up so much time.
That's okay. Well, I mean, if it's helpful, then just, I don't mind doing it.
Yeah, it was very helpful.
Okay, thanks. There are a couple of things I do want to talk about though. Your final exam, I'm going to move it. I had mentioned, I'm thinking, is December 6th the last day of exam or is it last week? I mean, last day of classes is December 6th? Does anyone know? So what I'm thinking is I'll move the final exam to the last class. Does that work? Because I have to include all the portion that I went through in the exam, because if I have the exam first and then have some portions covered after, then they won't be included, which means.
Yes, December 6th is our last class.
Okay, so that's when the final exam will be. Does that work?
You mean the second midterm, right?
Yes, second term test, yes, sorry. No, the term test too, I meant. Yeah. Yeah. Mm-hmm. Just the second part. Yeah. Yeah, I'll just post it on the your courses too. I'll just make that change in the syllabus so that that's kind of solidified. And then I do want to maybe set aside 1/2 class for Q&A like I did last time, but for your final project, because I want to make sure that I'm clarifying. I hope you already started working on your final project, because if you haven't, start now, because I am expecting fairly in-depth, new, latest literature, peer-reviewed literature, that is all the papers that are published in the last 10 or 15 years to include in your paper, and not anything super ancient or superficial. I'll post an example of what your final project, the project paper should look like. But would that be helpful to have that Q&A before the break or in the last week of classes?
Is the
Before
Q&A
break,
for
right?
the
That makes sense.
presentation?
Maybe the class before the break. Yeah, I posted it on your courses. Yeah, there is the outline. But I feel like, again, based on how the exam went, I don't know entirely where you stand, how many papers you've written, what your level of understanding is when I said paper. So I just want to get that clarified. So the class right before the fall break, we'll do a Q&A, come prepared with your questions to ask. And then I'll post an example paper that you can use as a reference. Okay, yeah, that's it for today. Thank you.